Hello, everyone. I'm Erva from the Biological Data Science Lab at Hacettepe University, Turkey, where I'm currently pursuing my PhD. And I'm really excited to be here today to share our project, ProtHGT, which is a heterogeneous graph transformer model designed for automated protein function prediction using biological knowledge graphs and language models. Okay, so I want to first start with a brief introduction to the protein function prediction problem. So as we all know, proteins drive and regulate essential cellular processes, and identifying their roles is key to understanding biological systems. And to make this process systematic and machine readable, we use biological ontologies like gene ontology, which provides standardized vocabularies for annotating protein functions. And while traditional wet lab experiments are accurate, they are also costly and time-consuming. And to address this, computational function prediction methods have emerged as faster and more scalable alternatives. And that said, these computational methods aren't perfect either. Many rely heavily on single data types, such as protein sequences, which fail to capture the full complexity of protein functions. So proteins actually perform or modify their roles through interactions with various biological entities, such as protein-protein or drug-target interactions. And to capture the complex functional characteristics, we need more integrative models that combine diverse data sources representing these interactions. And to integrate and represent these diverse relational data sets, we use knowledge graphs. These are advanced data structures where entities are represented as nodes, as you can see here, and their relationships are represented as edges, these connecting links. Now, to process these types of data sets, we turn to graph learning. Unlike classical machine learning, this enables models to learn directly from graph structures. So it captures latent patterns by utilizing both the relational connections and also their like the node's intrinsic features. And this approach can be applied to various prediction tasks actually. And in protein function prediction, this can be defined as a link prediction task. The task of predicting these unknown connections between like this one, but between protein nodes and functional term nodes. And in this research, we introduced ProtHGT, a heterogeneous graph learning model designed for gene ontology-based protein function prediction. And let me walk you through our workflow you see here. So we first created a comprehensive knowledge graph that integrates various entity types like proteins, pathways, and compounds, and the relationships between these. And we also incorporated vector representations of additional features of entities, such as like protein sequence embeddings and also embeddings based on Go term hierarchies to better represent the characteristics of each entity. And to process this rich data set, we use the heterogeneous graph transformer network, as you can see here. This approach allows the model to capture the unique characteristics of different node and edge types within the knowledge graph and learn meaningful representations and patterns. And I'll explain each step in more detail in the following slides. So our graph data set includes 11 different node types as listed here, along with their data sources. Most of these were obtained from the crossbar system, which integrates biological data from various databases and generates user query-specific knowledge graphs. For this study, we extracted all interactions that involves all UniProt KB Swiss prot proteins. And by the way, if you'd like to learn more about the crossbar system, please visit our poster, or you can join my talk tomorrow at 12:20 in room 3A. And we would be happy to share more details. And at the time, we created our input data set. Functional terms, domains, and EC numbers weren't available in crossbar, so we integrated them from their own databases because of their importance in determining protein functions. And here you can see all the entity types and their relationships within the KG, And finally, to enhance predictive accuracy, we incorporated these feature vectors for each entity type using embedding methods. Usually they are protein or chemical language models that are used to represent sequences of proteins or other entities as numerical vectors. And we experimented with 3 different protein language model embeddings for proteins. And these are ESM2, ProtT5, and TAPE to investigate their impact on the performance. So here you can see the node and edge type statistics of our data set. We have a total of more than 500,000 nodes and nearly 4 million edges. And to ensure a reliable model evaluation, we applied UniRef 90 clustering to only keep one of the proteins that have 90% sequence similarity, and we aim to avoid bias between the training and testing sets by doing this. And we then performed an 80-10-10 train validation test split, and the protein function pair counts in each split are shown in this table. And finally, to create a realistic scenario, we generated 100 negative samples for each positive protein function pair. And we do this because, this reflects the fact that proteins are usually annotated with only a few functions, while most of the other functions need to be predicted as negative. So here is the heterogeneous graph transformer architecture that we used for the high precision function prediction. This HGT is designed specifically for handling heterogeneous graph data sets. And unlike traditional GNNs that treat all nodes and edge types the same, HGT actually processes each node and edge type separately by introducing type-specific parameters to preserve the unique features of each relationship. It also uses an attention mechanism similar to the original transformer, of course, to focus on the most relevant relationships for the prediction task. And we trained separate models for each Go category because the terms within these categories show semantic and hierarchical differences. As you can see here, each model is a neural network that incorporates many HGT layers to process the heterogeneous graph. And through these layers, the embeddings of the protein and Go term nodes are updated and then passed to a multi-layer perceptron that outputs the probabilities for each protein function pair in the data sets. So to evaluate our model's performance, we compared it with a wide range of other methods using the time-based split data set from the ProtHGT study. Here below you can see the timelines for the training and testing annotations. And in the bar plots, our model results are shown in red. We trained a total of 9 ProtHGT variants using 3 PLMs across the 3 training tasks for each Go category. And as you can see in this plot, these are Fmax values, by the way, so higher is better. Our model consistently outperforms all other methods across all 3 categories. And the comparison includes other heterogeneous graph models such as DeepHGT and PSP-Go, which also perform well, but they model a narrower range of relationships compared to our graph, mostly limited to protein-protein and protein function links. We also compared it with sequence-based models like pFresco, TAIL, and DeepFry, many of which also use language model embeddings, but they lag behind heterogeneous graph-based methods. And among the different ProtHGT variants, the one using the ProtT5 embeddings performed best overall. And but all 3 were competitive, and this shows that while high quality initial embeddings help, the strength of the rich relational graph modeling is key to our model's success. So we also compared our models to their corresponding base PLMs using raw embeddings from ProtT5, ESM2, and TAPE. And for this, we used the PROP. This is a benchmarking framework developed by our lab, to test how well protein representation models capture functional properties. It actually includes a public leaderboard where researchers can submit their own models and compare results against baselines, including both traditional representation methods and also modern protein language models. And in our experiments, we gave probe the protein embeddings produced by the final HGT layers of our models, trained separately for each Go category, of course. And then probe processes these embeddings using simple classifiers like SVMs to generate predictions on its test proteins. And as shown in these figures, all ProtHGT variants achieved performance comparable to or better than their PLMs. And this highlights the added value of combining PLM embeddings with heterogeneous biological graphs. Here, an interesting result is that our model trained on the biological process terms performed best, not only on the biological process tasks, but across all three categories. And this likely reflects that the fact that biological process terms represents a broader and more diverse set of biological functions, and the training set is also larger for the biological process. And that gives the model a better chance to learn generalizable protein embeddings, which then benefit the downstream prediction tasks for other categories as well. And to assess the role of different node types in our heterogeneous graph, we performed two ablation studies. Here, I'll focus on the results for biological process prediction due to the time limit. So in the first analysis shown in the top panel, we removed one node type and their associated ages at a time. And as expected, performance was highest when all node types were included. However, removing pathway and molecular function nodes caused the biggest performance drop. So in contrast, removing node types like compounds, domains, or phenotypes had little effect, suggesting they contribute less directly to this task. And in the second analysis, shown in the bottom panel, we trained models using only one node type at a time and their relationships. Again, using all node types worked best, but using only EC numbers or pathway nodes still show still gave strong results. And so overall, both analyses support this key takeaway. Using a diverse set of node types leads to a better and more generalizable prediction set. And finally, we performed a use case study to validate the biological relevance of our model's predictions on a few selected proteins. And in this slide, we will focus on the keratin-associated protein 23.1. Here we can see selected predictions for this protein. Our model accurately predicted the manually created annotations for protein binding and cytosol with a high probability. And additionally, the model output another high probability annotation, which is the term structural consonant of bone. So the relationship between this term and the protein is not currently reflected in existing data sources. So this prediction was evaluated as a false positive. And to investigate which connections resulted in this prediction, we checked the underlying graph, as you can see here, where we see multiple connections between the keratin-associated protein and bone-related functions. So, for example, the protein is linked to pathways like keratinization and developmental biology, both associated with bone-related processes. And it also connects to some proteins, you can see here, which share manually created Go annotations and participates in overlapping pathways with our protein. And some proteins that also take part in developmental biology pathway also contains the same domains to our query protein, highlighting this highlights are indirect but like biologically meaningful associations that likely contribute to the prediction. Here, I want to note that I'm not focusing on this example to claim the predicted function is definitely biologically relevant or correct, but rather to highlight how ProtHGT enables interpretability. So by tracing the connections through the underlying graph, we can validate known functions as well as exploring unexpected functional links and generating new hypotheses. Okay, so to make ProtHGT accessible to a broader range of researchers, we built a web-based tool that requires no local installation or programming skills. You can see the screenshots from that here. So as you can see, users can input UniProt IDs by search or by upload, choose a Go category, and then run predictions. And the results are very easy to browse and filter. And the filtered predictions can be exported as CSV files for further analysis. And beyond prediction, our tool also enables interpretability again with the View Knowledge Graph tab here. This shows the local graph around the protein, letting users explore why a prediction was made. And this subgraph is also downloadable for further analysis. So I want to note that currently ProtHGT supports predictions only for proteins already in the KG. So we are working on a new feature that will allow predictions for novel proteins by building their graphs on the fly using data from external sources, and this will make it possible to analyze new proteins directly from their sequences. Okay. To conclude, I want to note that what makes ProtHGT even more promising is its adaptability for future enhancements and diverse applications. It can easily integrate new data sources and larger data sets to uncover novel biological insights. And finally, you can check our full text preprint by scanning this code. And ProtHGT is also available both as a programmatic tool and user-friendly web server accessible via the codes shown here. So I would like to thank our group, especially our PI, Dr. Doğan, for their support and contributions. And our lab is presenting a wide range of exciting work at this conference, and we would be happy to have you join our other sessions. Thank you for your attention.
Thank you. This is very interesting. I wanted to ask, I'm not familiar with this type of method, but if you work with different modalities, you will have different coverage. Some will be rich and some will be very sparse. How does this method handle this?
I can't really hear the question. Can you
So some methods are you use different modalities. So some methods have rich coverage and others are much more sparse. So how does this method handle this?
The modalities in our the workflow is only the interactions between the biological entities. I don't know if you're asking about this one, if I understand the question correctly.
So you build a collection based on different modalities. So you bring the embeddings from different types of experiments, different types of data. Some are very rich. You have a very rich coverage of the connections, and others
I get that. So we try to choose the modality type that has the widest coverage. So, for example, for proteins, we chose the sequences, and like for compounds, we also choose their smiles or like selfies embedded notations. So we choose the widest coverage ones. Thank you.
Hi, great talk. So I was wondering if it would be possible to make the language model like representation, because it's a pooled representation, yes?
Yeah.
Across the sequence, if you could make that learnable in the context of coding. Because if you employ it as a learnable embedding, you then be able to basically infuse that embedding with the knowledge graph information. So for downstream inference, you could just use the updated weights of the. It's like you're almost fine tuning with the knowledge graph in mind.
Yeah, but within our workflow, we are just it can be named as fine tuning. But if you fine-tune the embeddings, initial embeddings using our workflow, then they can be only used for function prediction. If you are looking for something like that, then it can be done.
Yeah, that makes sense. I just wonder if in the actual, like, because you're using the frozen embeddings.
Yeah.
Right? And so after you've taken the frozen embeddings, have you looked at the embeddings if they change, if you make them learnable after the context of training on the knowledge graph?
Yes, so the final embeddings are, I don't know if they're comparable to initial embeddings because, yeah, I see what you mean now. So we compared the initial embedding space and the updated embedding space using actually dimensionality reduction techniques. And we saw some really serious like clustering after the updating process, but I couldn't include it in my presentation because of the time limits. And also we are actively working on that analysis. So we compared that, yes.
Fantastic.
Thank you.
The knowledge graph is really cool.
Thank you.
Thank you. I'm sorry if I missed this, but what happens to proteins that are new, like completely new?
Currently, we can't generate predictions for that because they usually, their interactions are usually doesn't included in the existing data sources. So if you have a new novel protein sequence, we currently cannot generate a prediction for that, but we are working on a feature that would take the sequence and then search it in sources like InterProScan, for example, to take their domain data, and then we can create the interactions between that protein and their domains, and also other data sources like this. And then we can, after we create the knowledge graph, we can generate predictions for it.
So it's enough to have a domain similarity?
No, it's not enough. There would be other data sources we would source the interaction data set. It's just an example. It would be enough just to have the sequence.
So, but if it's enough to have sequence, then a new protein can be predicted.
Yeah, it would be
So
possible.
what happens with a new protein?
Yes, so currently we didn't like implement that on-the-fly data retrieval process, so it is not currently available, but it will be in our future work.
Okay.
Thank you.
That brings us to the end of our session, so we'll take over all of the speakers on this session.